This panel, we've had a change of the moderator, and Mike Barrett will be the moderator instead of Kate Rubens, but Kate wanted to be able to speak more from the panel, and certainly she's one of the international experts in genetic studies and has conducted genetic studies on the International Space Station. Mike has been introduced several times. He's a long-term friend. Usually I use an illustration that involves horse manure when I introduce him. I'm going to skip that this time. But Mike's a colleague and a friend. He was a flight surgeon, and he's flown twice in space, a long-duration flight and a short-duration flight. He's headed the human research program for a few years, and he's been instrumental in arranging for panels and speakers and involved with the International Space Medicine Summit. It would be hard for it to come off without his expertise. Since we started this particular year, I've been very excited about this particular panel in genomics. There's so many opportunities to provide optimum care for our crews. We don't have, huge numbers. We have 4 a year. If we go to Mars, there'll be very few people. And the ability to do pharmacogenetics and optimize their medical care, for example, if we do genetics and they have a possible prolonged QT syndrome, we can avoid macrolide antibiotics. We can know what antidepressants work by using individualized medicine. And if we know things that might occur that even they're pre-symptomatic, we can work with crew members and put in preventive medicine programs and those sort of things. There are some downsides, of course, to some of the genetic studies. It's fairly early. They know a lot of things that you can pick up with genomics, but also you get some incidental findings, and it's kind of hard to know what to do with those. And also there are some ethical issues of this, and I'm sure Mike and the panel are going to explore many of these areas, and I'm very excited to hear what everyone has to say. Mike.
Okay. Thanks very much, Richard. I'm not a subject matter expert, and I'm not Kate Rubens, as Richard pointed out. I am definitely a consumer of genetics products and application to space medicine, so definitely have a keen interest in it. And just to start, I think Richard gave a superb introduction. I'm not going to repeat that. We're going to use the buzzword omics for basically everything along the central dogma chain of genetic expression and products. So genomics, transcriptomics, metabolomics, proteomics. We're just going to say omics or astroomics or something like that. I think again, Richard did a really good job of talking about how this might apply to us. And I would like the panel to be thinking about the ethical questions, because almost every article out there on applying genetic techniques to spaceflight is wrapped around this genetic issue. However, I'll throw out the first grenade before we start. Given the trajectory and where we think omics analysis might be in medicine in the next few years, I'd like you to think about the ethics of not applying genetics principles to space flyers to get a full understanding of the hazardous environment and what their human condition is as a result of it. Is this going to be like an MRI that we had and didn't use to take the brain image because we were afraid of release of that image? So I'm asking you to use your crystal ball a little bit as we look forward to the next few years and where this is going to be in mainstream medicine. So first, down the list, we got a great panel, actually. This is going to be a lot of fun. Kate Rubens, astronaut and veteran long-duration flyer, also chief of our EVA branch, by the way, one of our ace spacewalkers. I've spent a lot of time underwater in the pool with this lady laying cables for new docking adapters, so she's fairly multi-talented. But also the first to perform DNA sequencing off the planet. So we'd like her to comment on what that has been like. And then I have a kind of a special question for her afterwards, so you can anticipation.
All right, so really great to talk to you about this, and I think it's extremely exciting that we consider this kind of panel to be sort of almost a mandatory thing now. It's really an issue that over the last couple of years we've been talking a lot about, but I think it's going to be increasingly more at the forefront as we get more genomic information. So right now the state of things are that, we can sequence genomes. What we do with that, or the associated transcriptome or metabolome profiles, what we do with that information is dependent on what we know about the genome. And right now we know a little bit, like we can tell women that are going to be predisposed to breast cancer from a very specific set of mutations or a few things about cytochrome P450. But we actually can't predict a lot about human health and disease. So I think where genomics is right now is the fact that we should probably be starting to take this information. It's going to be really important to start getting this information for our naive space flyers, and we have a lot of those in the office right now. We've got a huge number of people that have not flown in space, but they are rapidly being assigned and flying in space. And so every time they then fly to space, they're no longer space flight naive. And so if we don't take those samples early, we miss out on that information. I'd also like to encourage us to think about genomics a little bit beyond just thinking about the human. And we're very interested in obviously astronaut health and human health and effects of spaceflight. But what we haven't done really with genomics is start looking at other things like model systems. So, a great experiment is just these really simple kind of evolution experiments in space where you have a chemostat and you're watching generations of yeast evolve and change, or looking at microbial changes during spaceflight, or even things like our rodent system or fly systems. And you can do those experiments very, very quickly and look at multiple generations and look at mutations and heritability, and then sort of long-term effects of spaceflight as you do many generations. And so I would think past just the human for genomic information. The other thing is I think we're very much in this mode, and this is a standard science mode of we've got a hypothesis, we go test it out, we take a few samples, and a lot of our studies involve, ends of 5 to 10 people, and we get maybe 5 to 10 samples per person. But I would say for the genomic studies, first of all, a lot of times we don't have a hypothesis. A lot of times it's logistics of the sample collection, and it's very hard to have a hypothesis about the action of 20,000 genes. And so really what you need to do is set up very robust and well-controlled data collection, and then you basically collect all the data. There is not much of a hypothesis. It's get the data, get huge amounts of data, and then you can take a look at what that data is doing and pull out various responses. And in order to do that well, we really need to pay attention to what I'm going to call the metadata that we collect. So you can take blood samples from a person, but if you don't know what they just ate or how many hours they slept or how disrupted their circadian rhythm is, or whether they're really stressed out because they've got an EVA the next day or it's a Saturday and they're just relaxing, you're not going to know what's influencing that gene expression. And so I think this is actually a really unique area where behavioral health and performance and the kind of med sample collection intersect. We want to know what's going on with people's sleep cycle, their circadian cycle. We've got the ability to track food now on board Space Station. We can tell how much they've had to eat and what they've had to eat, what they're drinking, what their timeline looks like, what their overall stress levels are. Then also combining that with environmental parameters. So what's their CO2 exposure? Are there other small trace contaminants that could be having some effect on the system? What is their timeline in terms of, are they in full homeostasis and 3 months into the mission, or have they just docked a space station and fluid is shifting everywhere? So all of this kind of metadata is what allows you to interpret the data. So I think we really need to think about in every genomic study, yes, you're collecting the genomic data, but how much information you have to associate with that is going to determine the quality of your ability to draw correlations and understand why the gene expression patterns are changing or why you're seeing epigenetic modifications. So really thinking about good metadata collection. And then also thinking about sources of variation. So these types of experiments are a little different than our normal experiments in that they really involve hundreds of thousands of data points. And with a normal lab experiment, you make some stimulus and then you look at, for example, a change in a single protein before and after you add some transcription factor. You're changing the system in a very specific way, and then you're assessing one part of that systematic response. With genomic studies, you're actually assessing how the entire system changes. And so the one thing I can guarantee you all is that if we do genomic studies on board Space Station, we will see a lot of changes. The question is what's driving those changes and how well have we defined the granularity of our data set to understand what's driving those changes. So a simple example is, let's say we want to do a transcription study and we take whole blood or PBMCs, which is a mixture of cell populations. Well, neutrophils have their own specific transcriptional component, which is very different from lymphocytes. So if the proportion of neutrophils changes even just 1%, it's going to look like we've turned on all these genes. It's not actually induced gene expression, it's just a change in your sample type. So it's really critical, and it's a little bit hard to do this on board Space Station, but it's really critical to think about how we process these samples. So how long have they been out of the body? At Stanford, we showed blood samples that had been from a blood bank for even 2 hours were having massive transcriptional changes. They're ex vivo at that point. So how long has it been out of the human body, which really argues for a lot of processing in situ on board station. We don't have to sequence it necessarily, but we at least need to get it to stasis. It's got to be frozen or fixed somehow. And then what's the component mixture? What are the cell types that we're assessing? Is this a mixed cell population? Do we have information about what cell populations are in there? So you're going to get this picture of changes, but really the validity of your experiment depends on how well you've defined all of the informational inputs into those changes. And have you lost information? So you can think about this like image compression, right? If you compress an image, you can't get back that information again. You've now gone to less information. And so when we collect these genomic samples, we need to collect all of the right information so we can reconstruct the sources of variation. And ideally really pinpoint it to what's true variation due to spaceflight versus what's variation due to just the fact that our bodies are amazing at responding to changes, and they will respond to basically anything, which is having a Snickers bar before you do a blood draw, or if you haven't slept, or if you are slightly changing your percentage of neutrophils. So the takeaway is I would encourage a lot of metadata collection and a thorough understanding of the sources of variation in our sample.
Okay, thanks very much, Kate. I'm going to hold my question for the sake of time, and let's go to Professor Penelope Bonin. She's Associate Professor of Human and Molecular Genetics at the Baylor College of Medicine, and she has leveraged her expertise in genetics, biotechnology, and bioinformatics to real-world translational research discoveries. So she's also serving as a advisor to the Human Research Program.
Thank you, and thank you for having me today. So I wanted to give you some brief comments on the use of omics both in the research setting and in the medical practice setting, and really want to frame that as two very different settings, and it's important to keep that in mind as you go forward. So with regard to the research setting and generation of omics data has been tremendously powerful in speeding the progress of discovery in the research setting, and then allowing us to translate that into medical practice. And when you do have a small n, as you do, and as we do when we study rare genetic disorders, which I study, one of the strategies that we have found to be very powerful is to use your omics as hypothesis generating. And keeping in mind everything that Kate just said about collecting the right data and understanding exactly what information you have is key for interpreting that. But when you have a small n, associations through omics data typically will not give you definitive answers. And so you generate hypotheses that way, and then you follow that with basic science to give you really, truly definitive understanding and answers that can be translated into your medical practice. And on the medical practice side of things, FDA has already approved lots of tests that involve omics data. They have been not just FDA approved, but they are CMS approved, which means that even Medicare and Medicaid are reimbursing for those tests. And that tells you sort of how well they are accepted in the practice of medicine and in their ability to be actionable and applied in responsible ways. So, for example, we have pharmacogenomics tests. The FDA has approved pharmacogenomics testing for 7 genes, and they affect multiple different drugs, the metabolism, and therefore the dose that a person would take of that drug. And that's something that could be applied to your population of interest. And there are also tests there are many tests with precision medicine that are involved with cancer treatment and prognostics. One point I wanted to make about that is you don't have to, in your medical practice, sequence a whole genome and interpret a whole genome in order to get the information about your pharmacogenomics genes. So if you wanted to, in your medical practice side of things, strictly interpret those genes for which we can definitively take action with 100% confidence, that can be done. So you don't have to mix these two sides of things, the research and the medical practice. You can separate them. And I think that NASA already collects personal and private information on each individual. And they already use that information, and they already have sort of protocols and policies around protecting the privacy of individuals. And so I think you have a framework that you can already start to utilize this information, or think about adding to that framework to utilize this type of information. Thank you.
Okay, thank you, Dr. Bono. All right, Leroy Chow, another consumer of potential long-term genetics testing and well known to everybody.
Right. Well, good morning, everyone, and it's great to be on this panel. I'm definitely the lay person here, but I am a user, and so I'd like to kind of share with you my personal story on this, and then make a few observations about the ethical implications and things like that, as far as from an astronaut standpoint. So I was a few years ago, Graham Scott of Baylor College of Medicine, back through the Center for Space Medicine, approached me with an interesting idea. He said, well, we'd like to take blood from you, do your genomics, and then get some samples from NASA from way back when, and see if anything's changed during, from the time you came into NASA to through your space flights. I said, well, that sounds like an interesting thing to do. And so I signed up for that, and then he went on with John Belmont, also of Baylor, back then, and also signed up several more of my colleagues and peers, folks who had left NASA, who agreed to be part of this research program. So it took a little while to get through all the hoops at Baylor, but then finally they got kicked off, and so I went down and gave blood. They took a few vials of blood, and then a little while later, a few years later, they finally had some results. And so it was very interesting because then I had the debrief, and fortunately for me, the results came back that I had no bad markers for any diseases or anything like that. So, I thought that was pretty good. And I went home and told my wife, and expecting her to be happy about it, and she said, oh, great, so what you're telling me is that anything that's wrong with our children is my fault. I hadn't thought about it that way. But anyway, so I didn't give too much more thought about it, but one of the most interesting parts of this research project was the ethical briefing before I agreed to sign up for it. So, sat down with John, and he took me through all the different things that they might find or implications of going through this testing, including back then the original plan, which they didn't do. They were going to also sequence our parents to kind of compare different differences there, or similarities and differences. He said, well, one of the things you might find out is that your father is not who you think, right? It's genetic testing, but I think my dad is who my dad was my father, because there's so much of him in my son that there's really no other way to explain it. But other implications that came up, he said, well, you may find these markers for cancers or for early onset Alzheimer's or things like that, and maybe you don't want to know. Well, I've been, I'm an engineer, I'm a pilot, and I'm an astronaut, so I always want to know the condition of my craft, which in this case was me, right? So I had no problem with that, and signed on the dotted line, and off we went. But then I thought a little more afterwards about the broader implications of this. There are a lot of science fiction movies out there that kind of talk about or touch on these themes. But there was a very real example that I thought about. And, I don't know if this is true or not, but at least one astronaut had alleged a few years ago that NASA had, without his approval, taken some of his archived blood and, done some of this kind of work on it. And so I don't know if that's true. I don't know if it was an accident. But if it is true, that seems to me to be a pretty big ethical lapse. Okay, and of course the concern that astronauts have, active astronauts, is that something the doctors find can and probably will be used against you, right, to disqualify you from future missions or anything like that. And so that's, kind of, brought up a little bit of unease. I know there was some discussion in the astronaut office about this new technology, this is some years ago, and how it should be applied. And that kind of goes for the broader topic of technology. Any technology that emerges and becomes developed, Of course, the whole thing is just because you can do something doesn't mean you should, right? And there are numerous examples in technology today that we could talk about. But, so that brought up, well, okay, if you retroactively apply something like this to disqualify people, maybe that's not a good idea. But if you go forward and you use it for selection, well, why shouldn't you? This is another tool you have to discriminate against who might be a good candidate and who might not be a good candidate. And so I think this technology really is exciting. I mean, we've seen just in the popular press examples of the potential of this technology, and I think it's exciting that Kate got to do these experiments aboard ISS, using a small device, and it's really come a long way. So as a user, I'm very excited about it, but I'm also, want to make sure we're cautious. Just like any other technology, we've got to make sure our ethics keep pace with the development of that new technology.
Okay, thanks very much, Leroy. All right, Dr. Bill Pulaski, 5-year program lead for the Human Research Program, and talking a little bit about HRP's genetic research and hopefully a little bit about Gene Lab, if possible, if Craig debriefed you on that.
He did.
Okay, excellent.
So thanks, Mike. I'm expecting, I guess, in the long term that omics will have a profound effect on how we do medicine during Mars missions. We're not there today, as Kate was saying, and Penelope. We're sort of in the Wild West stage of all this stuff, but things are starting to flesh out and to congeal, and I think we're going to have a lot of new information by the time we go to Mars, which I'm still guessing is probably 15 or 20 years off, that we'll be able to use to help to guide the way that we do crew health care during these long duration missions. So our first foray into this, interesting that you mentioned 5 years, when Mike wrote me into this job, he left me with a decision that I was unaware of, that the crew member that had been selected from the U.S. that had that was going to go on the 1-year mission had a twin brother. I had no idea. I'd been away from NASA for 5 years. And he said, we have to decide whether we're going to do some omics studies on these crew members. And he left that for me, and we decided, after talking with our scientists, that we should do that. And it wasn't just because we could get some omics data on 2 crew members, basically one that was on the ground and one that was in flight, but we had a huge problem with being able to do omics data on any crew member in flight because there were no policies at NASA, and we had to drive the policies. And these things were very complicated because there are a number of laws, and I think Leroy talked about how we might use omics data. There's a law that disallows any employer from using genetic information to modify the work that a crew member, in this case, or an employee does. It's called GINA, and it's a very serious law. Now, that doesn't preclude us using that during the selection process for new crew members, as I understand it. But we had to get past the GINA law. We also had to get past the issue, while astronauts are not in a protected class by the IRB rules, they are celebrities and they are stalked by people, and so we had to be very careful about privacy of their data and how we were going to maintain the privacy of the data. And then we had to cope with the problem that when you have genetic information from an individual, most of our studies just apply to that individual, but the genetic information now applies to what might be going on with the individual siblings, with their progeny, with their parents, with their entire family. And some of the sorts of things that we might find might make people in the U.S. system be ineligible for insurance coverage because of things that we found. And so we had to figure out how we were going to get by all those things. So we managed to do that through a lot of work and effort. There is a policy at NASA now, comes out of the Chief Medical Officer office, to be able to collect genetic data for research purposes. And we have had to go through a very difficult process of how we store it and make sure that we can maintain it in a private file that can't be hacked and that no one else can have access to except those that need to have access to it. So in the future, I think that the vision is that epigenetics and precision medicine and what we learned, some of the things we've heard about before, are going to drive our understanding of the stressors that crew members are being exposed to and what the response is to it. And the response of each individual could be different, so the treatment for each individual could be different based on the genetic predisposition of that person. So we're looking at what sorts of epigenetic markers might indicate an excess response to some kind of radiation stressor, for instance, or lack of sleep, or CO2, or something like that. And recognizing that, then the docs would have the capability to say, well, I think based on your history that we would treat it this way, whereas somebody else we might treat a little bit differently and suggest something different. So that's where we are in the future. Today, we're doing an awful lot of work trying to capture biomarkers. What are these markers, these epigenetic markers, or other omics markers that might indicate fairly clearly that there's some stress going on that we need to deal with, either giving the crew member back some information or having the docs deal with it. So we're doing a lot of that we're doing in model organisms. And my sister organization at NASA, the Space Biology Program, has established a very elaborate gene lab as a repository for all the genetic information we've got from model organisms from both HRP and space biology things. It's an open source, genelab.nasa.gov. And it's open source for anybody who wants to get to it. And I think there are data from 178 separate experiments now that are captured in there. So it's easy to find, and people that want to go and start understanding it. And as we do more experiments in space or in environments like our NASA Space Radiation Laboratory, where we expose mostly rodents to galactic cosmic rays, we capture genetic information and we put it into this database for people to go and look at afterwards. I think one of the key areas, and Kate touched on it, that we're looking at right now is we have a very large experiment that we're getting ready to start flying that will look at the microbiome of the built environment, that is of the station, and the microbiomes of the crew members that are aboard the station, and we'll be able to track those over a mission. So we'll be capturing genetic information from the microbiome, from the gut, mostly fecal samples, I'm guessing, from the people, and then analyzing those and analyzing the genetic material we get from the bugs that we take from swabs around, and start being able to put together a big data review of the interactions among crew members and between the crew members and the environment, and also the evolution of bugs that are in the environment. So I think we're starting to look at some of the things that Kate was suggesting. The ethics is always an issue, and trying to figure out how to make decisions based on what you know about genetic material is always going to be a challenge. I think it's really important that we move slowly but deliberately in the way we move through this. One of the things we learned from the twin study, as Leroy mentioned, is that counseling is critically important. Counseling crew members before they sign up for a study about what to expect, when the data come through, what might be found during the analysis, and then when the analysis is finished, whether the information should be released or not. So there are a number of places during the sequence of these kinds of studies that we do counseling for the crew members. Let's see. Yeah, I guess that's pretty much it. I guess the last piece is the twin study happened 2 years ago. I guess they finished. They came down. We finally had. It took a long time to get all the data pulled together. We had a group of investigators from around the country who did not do individual investigations. Even they were selected individual. We formed a team, and we had a team look at at not only the genetic information that we gathered, but also at the metadata that might have applied to that. It took a long time to do the analysis to get everybody on board. We submitted that some months ago to one of the most prestigious journals in the world. My understanding, and I'm going to look at Chuck Lloyd because I think he knows the answer to this, is that rather than having two peer reviewers like we usually have for a scientific paper, they had something on the order of 20 peer reviewers. Is that right, Chuck?
Hey, well, I'm not certain if they had 20 of them, but I can tell you that they had 7 with a significant number of comments back to them. probably 6 out of 7 of those were very positive.
Yeah. So this was a very complicated study, and even the journals are looking at it a different way from a standard submission that you might make to a journal because of the importance of it. We were in the final stages of making revisions, and we're expecting it to come out in January timeframe. And so I think that there'll probably be a lot of information, a lot that'll hit the airwaves when it comes out in January. So we'll finally see the results of the first big study that we've done on astronauts in space.
Okay, thanks very much, Bill. One quick footnote is that we had actually tried to bring genetic studies to human spaceflight several times before, an effort led by Dr. Smith Johnston, who's well known to be one of our more forward thinkers. The agency wasn't ready for that, kind of said that we don't know how to handle the information, it's too risky. The Genetic Information Non-Discrimination Act was passed in 2008. That actually made it harder for us to readdress this in 2010. We shamelessly used the twin study as a forcing function to get the agency compelled to write down policy on the heels of GINA, but policy that we could use that would make sense to us internally and operationally. And I would almost submit that was one of the best uses of the twin study, this low-end study. So, and Bill helped to shepherd that transition nicely. Dr. Neil Pellis, cell biologist emeritus of NASA, former director of the University Space Research Association, and a longtime NASA career and medical school faculty member.
Thanks for having me here. I am not, it's changed now. I'm not Jeff Sutton. He's back there if you want to talk to him. But I have a somewhat different approach to what's going on, and it doesn't mean it's a contradictory approach or anything. I spent most of my life studying cells, okay, and a shorter time with some bacteria. So these are small little creatures, and you wonder why would we even bother? But they're fundamental to, particularly cells, are fundamental to the functioning of your body. For all the tissues, everything that you are, okay, those cells are there. And the interesting thing about them is that when you take this and translate this from a terrestrial environment to space, in which one of the more interesting forces that changes while you're there is actually gravity. And it's interesting because as a scientist, you look at this and you look across terrestrial life, and terrestrial life for the last 4.8 billion years has never really had to deal, okay, in an evolutionary basis with a diminution in the gravitational force. We do have hyper-G events that occur, and you do have genes that are programmed to respond to that, but you don't have anything that's pre-programmed by evolution to actually respond to a decrease in the gravitational vector's force. So that being, set in stone for right now, it makes this an extremely fascinating area of science. And anybody says, well, why do you go to space? Why? Well, we say, well, it's a unique environment. So what? But what happens is when we take terrestrial life to microgravity, it does attempt to make changes. And interestingly, a lot of the first things that we found in the genetics, and not so much omics, but gene expression. Omics covers two things, basically. Primary sequence of your DNA, the bases in your DNA. That's the letter code that underwrites everything that you are. Okay, and the second part is which ones of those genes are turned on and which ones of them are turned off. And when you respond to an environment that you've been programmed to be able to respond to, you turn on the genes necessary to be able to thrive in that particular condition, whether you're a cell or you're a full organism. It doesn't matter. It's pretty much the same kind of story. So that in itself is an intellectual forcing function for us to actually really intensify our study of not only the human genetics, but terrestrial life genetics as we're in space, because it will be informing to this entire transition. When we go to Mars, okay, we're going to take living things with us besides ourselves. And we have to know how they're going to function in that environment, not only for a couple of days, because, yeah, we can take plants and grow them in microgravity, seed to seed. Can we do 50 generations? Because you're going to need 50 or 100 generations of lettuce or whatever you're growing you're going to try to eat. We don't know the answers to that. We should study these things in cell-based settings because your gastrointestinal epithelium will turn over 1,000 times during a Mars mission. We don't know the fidelity of that replication for that many times. We have no idea what it's going to be like because we're not programmed to adapt and do whatever's necessary. So we got to see what that means. And I don't know what it's going to mean. I mean, you get crazy and I can say, well, after 500 days, we're going to see people that are going to have some malabsorption because their epithelium has changed so much that they can't absorb. So all of these things that we're talking about right now generate an enormous amount of data. And it's got a new term now in science, big data, and it's got a new math, discipline associated with it that's teaching us how to handle big data. Because you've got this huge multi-matrix of numbers, and you're trying to find a fluence in it that's going to inform you about a specific thing that's going on. We know very few of these things, and that's going to take us a while. But I think the important part about it is that there's much more interest in it now than there ever has been. And that the external medical community, which is a much stronger driving force than we are here in the space program, is actually pushing very hard for a lot of this to take place as rapidly as possible. So I think that despite the fact we have some concerns about it, and there are legal concerns, I agree 100%, but we also know that there are genetic signatures for predispositions. We don't know all the predispositions, but Down syndrome, we know the genetic predisposition. You got that signature. That's a bad sign. Apert syndrome, which is synostosis, and there's a whole list of things like that besides the kinds of diseases we've been talking about, like Alzheimer's and that. So there are many, many subtle signatures that are in that code that we have to study and understand. And I think that. When you look at it, it's like the little kid that's out in back of the barn at his grandfather's farm. He's out there digging away in a big manure pile, and his grandfather comes out and said, he said, "What are you doing?" He said, "Well, with all this manure, Grandpa, there's got to be a pony in here somewhere." Okay? And that's what we do with big data right now, and that's why the people who are accustomed to traditional hypothesis testing research are extremely, extremely dismayed with the fact that we're going to make this big pile and we're going to find a horse or a pony in there, right? But the mathematicians, I think, will save us. Honestly, I think they can actually put that to good work, and we'll be able to get the kind of information that we need for some of the problems that we face. I had a lot of things I want to say, but you got to be careful about when they talk about genes change in space. There are kinds of changes that can happen. You can have mutational changes because there are a lot of things. We have a natural mutation rate on the ground of 10 to the minus 5. That means 1 in every 100,000 times it's going to be a mutation that occurs. Right? That's very low. And we tolerate it. We've actually evolved in it, and we can handle it just fine. We don't know what that is when you get out into galactic cosmic radiation. We have no idea what that's going to be. We have some mathematical models that are helping us to try to understand it, but no idea as yet. So we've got a lot of work to do beforehand, and I think that with the diversity of the population we're dealing with, we can look at any one or two people that we want to, but we've got to be careful how far we extrapolate it, because we don't know all the things that happen. Because when you get a genetic change, as I told you, it could either be in the sequence or in the expression, but these can be just incidental changes with no consequence whatsoever. Others can be consequential and have real effects that you didn't even think about. And some of them are changes that occur and they don't affect you for a long time. So when we look at a snapshot in a given moment in space, that's all it is. It's a freeze frame, because it's a dynamic process that continues to go on until it reaches an asymptote for the environment that you're in. Okay? And that's evolution is what it is. We were brought to certain asymptotes, and that's what we are, and we're going to be what we are until something perturbs it again. And so we've got to get that dissected out so we understand whether or not there are changes that are occurring temporally that we are unaware of because we've been taking snapshots and not movies. Okay? Movies makes the data even bigger. So the problem, I'm not trying to paint this as an impossible problem. It's not. It's just a monumental problem that requires some very sophisticated assistance in getting through it. So I'm not going to go on. I got other things here, that we could talk about and you could ask me about. But there are some, unique things about radiation that really titillate me when it comes to thinking about scientifically how this occurs. We've got an organism called Dinococcus Radiodurants. Okay? You should be able to derive from that name what it is. It's very resistant to radiation. It's 4,000 times more resistant than human beings are. How did it get this way? We've never had that level of radiation on the planet. Okay, and at first we think, well, it's a visitor. But in analyzing, the preliminary analysis says that it has the ability to co-opt genes from other species that help them do this. There's another organism called Tardigradia. Okay, these are water bears. They're small 1 millimeter animals that have 8 legs. They tried to make them like they were insects, but they're not. They're something on their own. And they're very, very resistant to radiation, 6,000 times more resistant than we are. They're more resistant to desiccation. They're more resistant to thermal. Okay, whether you're talking about taking them down to a few tenths above Kelvin, or you're taking them into the range of 250 to 300 degrees Fahrenheit. They resist them. They go into a dormant state and they kind of hang out, and they'll hang out for 150 or 200 years. You throw them in water and boom, they come back in about an hour and a half and they're swimming around. And so there are a lot of things that we can learn. But obviously there are ways to be resistant to radiation that maybe we've never understood before. And I'm not presuming that we're going to genetically modify people to do that, but who knows, 150 years from now when they're really looking at a Starship mission, 100-year Starship mission, we might have people designed to actually withstand the aridity of what they have to go through. So with that, I'll stop. But that's the kind of stuff that keeps me up at night and off the street at night.
All right. Thanks, Dr. Pellis. Those are great comments. Dr. Virginia Wotring, Ginger to us, has been studying pharmacology of spaceflight for the better part of 10 years at Johnson Space Center and at the Baylor College of Medicine. She was a former chief scientist for the Translational Research Institute. And she's used all methods from paper questionnaires for astronauts, what drugs did you take in space and how do they work, to genomic techniques. So I'd like her to talk a little bit about pharmacokinetics and pharmacology in spaceflight vis-a-vis genetics.
Thank you, Mike. I'm very happy to be here. I have to agree with the previous panelists on comments about how we should be building the information that we're collecting. I think omics techniques will let us really grow what knowledge we have so that we can discover new things. Like Neil was saying, we need to take movies rather than snapshots. I think that high temporal resolution omics kinds of studies can help us build this sort of database from which we can make discoveries that could become useful for treating astronauts in the future, for diagnosing any conditions, maybe even before they happen. These are incredibly powerful tools that are being developed on Earth that we could be using in space. Now, there are certain ethical ramifications, and it's good that we're having these kinds of discussions now to sort this out and handle things properly so that the subjects and the medical practitioners can feel confident that data are being used appropriately. But I'd like to talk today about things we could do today. Dr. Bonin brought up some of the tests that are available now. I've been tracking this for some years now. I started doing this when I was working in pharmacology at Johnson Space Center. I was working closely with the pharmacy there, primarily Tina Bayous. We know what drugs are carried on the space station, and we also have some good ideas about what medications are actually used by the astronauts. When these genetic tests started being formulated, they were driven by commercial needs. And there's a big driving market for cancer treatments and treatments for major chronic diseases. Astronauts don't have major chronic diseases in general. So the test, it was an interesting intellectual exercise at that point. We learned about the kinds of SNP tests that are available. You can have commercial panels done from a blood test, cost a few $100s. It would help a physician decide which oncology drug to use for a particular patient and their specific tumor, which drug and at what dose that would be best, both safe and effective for them. And that gave us a lot of hope about, where medicine is going in the future. Well, I updated this information. Dr. Bayous and I are actually writing an article at the moment on this topic. And I've recently updated this information, and we now have a number of FDA-approved tests on the market now in the United States that are applicable to drugs that are carried on board the space station and, in some cases, used by astronauts. And these are in the categories of antibiotics, meclizine, which gets used for motion sickness from time to time, beta blockers, statins, proton pump inhibitors. These are relatively common drugs. And as I said, these are SNP assays. These tests are examining a very tiny portion of DNA. It's not whole genome sequencing. Doing this test is not going to reveal paternity issues or diagnose any problem you might be passing on to your children. There's really nothing newsworthy that comes out of the results of these tests. Frankly, no one cares if you have a valine instead of an alanine and some cytochrome P450 enzyme. That's not very exciting. But for a prescribing physician, that can make all the difference when it comes to what drug you are given and what dose it's given at. These tests come with clinical practice guidelines to help a physician use them appropriately. And there are actually people in this room right now who have disclosed to me that they have benefited from these kinds of tests. These do get used right now in clinical practice in the United States. And I think that they should be used at the Johnson Space Center for treating the astronauts as well. We've been talking about ethics and protecting the astronauts. This is a case where ethically we need to be using every tool we have to treat them as well as we possibly can. So I think there are two sides to that ethical coin. And personally, if I develop a need for a proton pump inhibitor, I want to have the test done before we decide what drug I'm taking and what dose I'm taking. And I hope that the same can happen for Kate and for Mike.
Okay, appreciate it. Thank you. Well, that's a good segue. Dr. Thomas Williams is a clinical psychologist and also a Master of Strategic Studies from the U.S. Army War College. And he has been, for the last few years, our lead element scientist for the Human Research Program's Behavior Health and Performance Element. He's currently engaged in a study on radiation and CNS effects, something our spouses are very keen to use as an understanding tool as to why we're so absent-minded when we get back from flight. Most of us think we're baseline. But I have asked Tom to talk specifically about some of the ethical problems. So go ahead.
Great. Thanks, Mike, and thanks for inviting me to participate. So one of the things we've heard in the discussions of each of the panel members is that there are clues to our past, our present, and our future health that sits within our genome. And how we tap into that makes a huge difference in our life, and it can have many positive and many negative consequences if we don't understand it. And the best way to characterize that is probably to say we have some genomic uncertainty, because we've characterized as of 2018 about 18,000 genes. So there's a little uncertainty there as to what gets manifested beyond that. And when you think about where we've heard medicine, it plays a role in that. We've also heard we use it for screening to determine whether medicine needs to be used. We've also, on the market, you can get genetic screening for the type of diet that might work best for you. And you can also find out if you have a distant cousin who may have engaged in criminal activity that now is forensically linked into your genome. because of traces of their DNA that they left at a crime scene, that because you've posted your DNA, they're now able to kind of forensically backward assess who's this person linked to. And in fact, several cases have been solved recently. So there are far-reaching implications when we talk about the genome, and it is sort of a new frontier for us as we look ahead. And it's important that we think about that in terms of, as we've heard, there are interactions of our gene and our environment. And the stressors may alter that genomic structure in some way because of the upregulation and downregulation of the genes that get activated as a result of the stress. And to do a holistic investigation of how that all comes together, we're going to have to link personally identifying information somehow to find out in what different ways has this individual been interacting with their environment. So that has some far-reaching implications. And because of that, the Office of Human Research in 2017 has revised the federal regulations with regard to human subjects research and the informed consent that needs to be provided. And that, it's called, interestingly enough, the final rule. Remember, we're all familiar with the common rule. Now this is going to be the final rule, which has implications for us perhaps. But that is intended to go in effect in 2019. And some of the considerations to consider with regard to that is whether or not a biospecimen is going to receive the same protections as a human subject. And so and the other aspect of that is they are bringing in what's known as a broad consent, so that there's going to need to be a different way that you consent when you're dealing with your genome, because you're going to need a range of choices regarding the consent that you're providing, including what's the storage and future use of your specimens that you're providing, and whether or not parents can give up the DNA of their children and later have the implications of that, and that, again, far-reaching. In particular, because of that broad consent, because you can link currently academic achievement to your genome, sexual risk behaviors to your genome, substance abuse vulnerabilities, and you can look at, in the psychiatric area, internalizing and externalizing disorders that are linked to your genome. So when we start to look at the manner in which we can reach into that future status that you may hold, it has pretty significant implications. So while the final rule modernizes that federal regulation about informed consent, there's 3 key preconditions that now need to be addressed. The information disclosure. What if we have genomic uncertainty? How can we get true informed consent, and how do we work toward that? How can we make sure that, given the complexity, that participants will truly comprehend what it is they're consenting to? And then how voluntary is it if parts of it are placed by someone related to you that can be then interpreted as applying to you? And so importantly, as we move forward, as looking at the federal regulations, there was an appeal that they not go into effect because of an important definition. The definition for a clinical trial, which accordingly will be defined as any intervention or evaluation that affects a biomedical or behavioral outcome. So that would cover just about all research that we do, and that would now be considered a clinical trial. But because of the full implications of that, Congress passed an omnibus bill to put that on hold to let the full impact of that be assessed. So I'll just finish by saying, as we think about the genome and think about the specimens, how and for how long will the specimens be stored? If and when will they be destroyed? How do we de-identify them when the participant asks us to de-identify them? And what are the risks of identity linkage? And we heard part of that, the nature of the personal information that's going to be disclosed about us, and what's the rationale for doing so? What's the opportunities to withdraw from the study if your genome is already out there? And then what's the possibility, and we heard some of this alluded to by panel members, the implications of the biological basis of what you thought your family system was, and the determination that that may be found to not be the case. So when we think about it, there's genomic uncertainty, and it's important as we move ahead to think about the context in which it gets applied and the protections that need to be placed. Thank you.
Okay, appreciate it. Great comments. We'll end with Dr. Kotov. Dr. Kotov, I think, is well known to all, the former flight surgeon, very long duration flight experience, 500 days plus, and currently the deputy director of the Institute of Biomedical Problems in Moscow. And I've asked him to talk a little bit about the genetic research, the omics research that IBMP is undertaking now. And if you would, if there's any relevance to the BION project and omics research. This is, again, the high altitude 800 to 1,000 kilometer satellite study.
Yes, again, so first of all, I really understand that I really not much know about this genetic as well. So again, this is good. And but I understood from all my colleagues from here on the panel, and as well as a result of our research institute. So how is interesting is this problem? How is which is a huge perspective we have, and how is it powerful is this science? And I'd like to say, mostly pointed on, so we are faced to the next stage of space medicine. I'm talking about the expertise space medicine. If you remember, we started with the morphology space medicine. So we select the crew members and assign them to the crew using all the data we got from the morphology researchers. Now with space medicine mostly working as a functionality. Physiology, functionality, and how the body, according to our knowledge about the space flights, about the physiology in space and weightlessness, we understand and select the crew members and select the astronauts according to our knowledge at functional level of expertise. But now we're faced really in the next level of space medicine. I'm talking about genetics. So it's very interesting, but very dangerous on another side, because as the previous speaker told us, so that's really we have a lawyer problem and law. We have an ethic problem with that, and but we need to figure out how to implement our knowledge, our researchers, our results of our science to real space medicine. It should be very careful in this area. And because the omics researchers open the new area of human functionality or any biological object functionality, and that's why during the next unmanned mission to space, I'm talking about beyond satellites, one of the main area of our interest is going to be G research. So that's why, again, and so I just call you to be more careful with this science and to be it's very interesting. It's very perspective, but please do it together. Okay, thanks.
Okay, proceed with caution. And I'll point out that the IBMP was the first to publish proteomic studies on cosmonauts a few years ago, looking at expression products pre-flight and post-flight, and really helping to establish this database. So, all right, I personally have a lot of questions, but we have 15 minutes. I bet you guys do too. So I see you're first, and if we can find our roving microphones. If not, just stand and use your outside voice.
So I've got a good voice.
Okay.
So great comments by the panel. I've had really good discussions with a lot of you on this topic. I've had the pleasure of sitting in on a couple of workshops over the last few years focused focus on this topic, so it's great to see the progression that's coming along on this topic, and the embracing of the community when it comes to omics in general. So I've talked to a number of you in this room about this, and I know a number of you work with the Department of Defense, but I come from the DoD perspective. So I sit on the Biotechnologies for Health and Performance Council, and so specifically we have groups that look at the ethical considerations, the legal considerations, and then also what's the latest and greatest technology and how it can be applied to the DoD. And so I'll tell you, and I know I've again had this conversation many times, but the DoD has a lot of the same considerations that you guys have been discussing with respect to the ethical considerations of if we know this is something we should be testing and we're not, what does that implicate? The privacy issues, so not so much that they're celebrities, but they're people, there could be people out there who want to use that genetic information for malignant purposes. Longitudinal monitoring, so that comes up over and over again, is when do they start to collect this information when it comes to longitudinal data collection, and I know that came up yesterday in the panel discussion. And then also the selection question. So right now they're not even touching that, but there's that question of do they start looking at this for selection information? And so right now, just as far as talking about, like Ginger and Penelope have talked about looking at what you can embrace at this moment and not looking at the full genome, but just looking at specific targets. So we're looking at low-hanging fruit, targetable approaches that we know are going to optimize health and resilience in these environments, and we think of it as less precision wellness, or but more like precision resilience when we're talking about individuals who are operating in extreme environments. And so things like genetic status that influence vitamin D uptake and synthesis, or things like omega-3 uptake or synthesis. So things along those lines where we know they're going to have huge implications, but it's not really going to have an ethical impact if you start measuring these things. So they're starting to take that into account in some of the elite military units just to try to optimize their overall health and wellness. And last but not least, Kate was talking about the well-designed studies, and especially when you're looking at transient measures. So things like transcriptomics. One of the things that we found in doing some studies with Navy divers is that one of the biggest results of that study was actually time of day, and it doesn't seem like it would have a huge implication, but we were doing gene expression samples that were at different times of day because we're doing pre versus post measures, and just 9 a.m. versus 5 p.m. had a huge drive and impact on overall gene expression signatures. So just something else to take into account as we're looking at some of these transient measures. And with that, I was just going to ask if you guys had any kind of crossover when it comes to some of these targetable approaches with the DoD, and that's it.
Thanks, Dr. Knaes. Okay, let's move the microphone up there and someone address Dr. Knaes' question. Any crossovers with DoD studies, and frankly, to spin that a little bit differently, when we're looking at things like the final rule, are there populations that perhaps should be exempt somehow from some of these blanket policies due to their unique
The military
circumstances?
is exempt from GINA.
Well,
The military is exempt
okay.
from GINA, and NASA and the astronaut corps is not. So astronaut corps is still governed by GINA, meaning that genetic information cannot be utilized in the selection process.
right. And I was thinking a little bit more along the research lines. And does final rule map to GINA? If it does, I missed that. No. Okay.
Someone said no?
I heard a no. Well, and we don't have to have a jurisprudence discussion necessarily, but
Separate. They're two different
right.
components. But the final rule does apply to us. It's a federal reg that
And-
applies to all human subject research.
Okay. And the question is should it in all cases? Should it?
NASA also has their own as well, so there's a different federal reg that comes from the CFR 45. And so Ochimo actually, our chief medical officer, signs off on that. And we have it's a little bit more specific to us. And so any exceptions or anything that's done differently is published in that CFR.
Okay. I'm going to make a command decision. Dr. Knaes is joining us at JSC starting in January, I believe. Visiting scientist part-time. So she'll have, she asked a very big question. So we're going to all benefit from that discussion that hopefully answers your question. And go up here to the back.
Yeah, quick question. I was a flight surgeon for 15 years, and I've been working
That's an
on
understatement. But.
I've been working on trying to write some Mars scenarios for medical care, and I did one in pancreatitis. And as I looked through all this, I found out that there's all these nasty genes that could give you a risk of pancreatic cancer by a factor of 20,000. And so I started looking at all these other diseases, and they all have these genes. And so I'm thinking, well, I probably don't want that code to go to Mars. But then I started to add up all these other bad genes that everybody's getting. And I finally, we like to select the best and get the worst of the best and toss them out. But as we start to add up every individual and all the nasty genes they have, they're probably going to have hundreds, maybe thousands of these really bad genes. So now we're at the situation where we have to pick the best of the worst. And I think that's where we're going here, is that, we still have to pick some piece of protoplasm to go to Mars. And it seems that we all are flawed in some certain way, and that our firmware is our firmware. So it could be that this is the perfect candidate, but we think you're going to get pancreatic cancer, so go sit in the car and don't play with the radio. We're going to pick somebody else. And it seems to me that there's going to be a competition to figure out what is the best of the worst profile to send to Mars. I thought maybe you might want to comment on that.
I'd like to give a little bit of information around that. As a human geneticist and someone who participates in clinical genetic testing, there are roughly 5,000 genes out of the 20,000 where we have definitively, conclusively been able to discover that if you have a mutation in one of those 5,000 genes, you will have a disease. And the genetics community, we call that penetrance. If you have the mutation, you will manifest the disease. There are about 600 genes out of the 20,000 that we have information that we feel confident about, where we can say, if you have a mutation in that gene, you will have some increased risk for a disease. That's not the one-to-one. That's if you have it, you may have a 30% chance instead of the population risk, which is 7% chance. So I would argue that we're not all littered with disease-causing mutations. I would argue that genetic disease, as we can be confident with the fact that you will absolutely manifest it, is fairly rare. And a lot of the genetic testing that is FDA approved is really actually based on transcriptomics. It's not based on your constitutional germline DNA. It's based on some of the transcriptional changes that happen. Typically, these tests are in the realm of cancer. And they're actually being done on your tumor RNA, not your constitutional germline DNA. And those tests are very helpful with understanding which drugs. There are 30 FDA-approved drugs that are informed by these transcriptional genetic tests that are done on tumors that are FDA-approved. And I think that those tests can be very helpful for NASA's implementation of the TREAT Act, for example.
Yeah, and I'd like to add, a lot of what you're talking about is genetic studies from the general medical area that are done on very diseased people. And so they're looking at people who already have cancer or serious cardiovascular disease or something like that. What we're looking at is actually a population of individuals that have already been selected to be quite healthy. And so if you're already in a place where you are probably selecting out a good number of these diseases that would give severe disease in their 30s or 40s. It is interesting to think about, hey, can we predict disease in the future? And obviously, if somebody's going to develop a serious condition on Mars, you don't want to select for that. But we have to look at these studies. Often will say, hey, there is a slight increased risk. We're not predisposed in this binary sense. And so if you have somebody who is a great astronaut and they have a 32% increased risk for disease versus a 28% increase of risk for disease, are we really not going to send them to Mars? So we have to look at not just the fact that a test exists, but how well that test predicts your likelihood of developing, and also what that time span of development is. Is this the fact that maybe you're going to get pancreatic cancer by the time that you're 60 or 70? Well, we probably are not sending 70-year-olds to Mars. So we are looking at us already selected healthy people who hopefully are pretty disease-free as determined by our flight surgeons. And we're looking at a very narrow window where they are still in a very healthy period of their life. So I think we'll probably be using this for some known conditions to select out a small number of known conditions. And we'll mostly be using it for helpful purposes where we're saying, how do they best metabolize the drugs, or what drugs should we send? Or in the case of them developing a condition, how would we treat this? So I think it's actually probably a very small number of people that would be selected out from a Mars mission, given that they're already in a state of health and they're fairly young.
I have one more thing to add, and this is a piece of information to give you more context about the numbers and the levels of risk. The American College of Medical Genetics and Genomics developed a list of 65 genes that if you have a mutation in that gene, you will absolutely have the disease. So they are 100% predictive, and they are adult onset. Those 65 genes are what we have as a community decided must be reported as incidental findings. And so if you sequence somebody for an indication of, a pediatric onset disease, but you find a BRCA1 mutation, you have an ethical obligation to report that because there are implications for family members, et cetera. The way that those 65 genes were decided upon is because they had a 100% predictive power, but they were also treatable conditions. Half of them are cancer related, half of them are heart disease related. And these are the long QTs, the familial CADs, and then of course the familial cancers. And I think those are the 65 genes that would be sort of the more relevant to your maybe wanting to consider who flies or not. But Kate's points are really well taken. At what age is the onset of the disease, and what's the likelihood it's going to be onset in the specific time that is relevant to your flight? But also the CADs and the long QTs. I mean, those are essential for giving somebody proper medical care and giving them the best medicine that we have available to us. And I think that we should take that responsibility and give astronauts the best health care that we possibly can.
Okay. And I would just, we like to frame that as you can take a new information and you can use to select, or you can just fly informed. And there's a big difference between those two, and that has to be done very judiciously. Guillaume.
Yeah, I would like to come back to something which has been said by Dr. Vautring about the pharmacogenetics and the testing for medication which are currently used in space. I was telling you last year that we had started a study on the ESA astronauts, testing them on the medication which are currently used on the ISS. So we have now concluded that study. And we have got very interesting results. It's as Kathy said, we are already dealing there with a group of very healthy people, and people. The idea was really not to use that for any kind of select out. It was purely on a therapeutic point of view to enhance and to optimize the way we can prescribe medication. We have seen, we cannot, because this study was purely medical and not research, we cannot publish the complete results. It's usually, that's mainly used for individual counseling. But we have seen huge variation, even for the drugs which are pretty common that we are using on the ISS. And we have very slow metabolizer, we have very quick metabolizer, and that has huge implication. And for drugs which are as usual, but could potentially be as toxic as acetaminophen, we have a huge variation. So this is something that I would really encourage to enlarge in the application. It's something which is not, as it was said, is not giving any indication on indication of potential diseases which might happen, but that's something which is purely practical, and that's really, really useful.
Okay, thank you, Guillaume. Yep. If there's one more, I think, Mark, you got your hand up first, and then we'll go here. And then I want to leave a good runway for the Apollo 50th anniversary. So first to Mark, and then we'll finish with our Chinese colleague.
Question for Kate and Penelope. For Kate and Penelope, how well do we understand genomic change in the control population? How well do we understand how the genome changes in a, quote, normal person on a month-to-month basis?
So, we have some idea about mutation rates. We've also got DNA repair mechanisms. And so there are large population-based studies that are oftentimes, most of these studies are focused around disease versus healthy states. So there are a few studies, for example, NIH is starting to expand. You may have heard of the 1 Million Genomes Project. And some of these studies have a longitudinal component. But the number of studies that really just study a purely healthy population and longitudinal changes over time are much less of a proportion than the disease versus non-disease states, because that's really what's driving the funding. People want to know, am I going to get cancer or cardiovascular disease, or if I do get cancer, what's the best cancer treatment for me? So just these basic studies that look at normal humans and how they're evolving usually fall into a little bit more of the basic research studies. And so there are some of those ongoing, but I would say it's maybe not as well understood or as well advanced. And there's also a lot of potential. There's been more studies, I would say, in terms of model organisms looking at yeast genome evolution. It's a smaller genome. It's a little easier to get a handle on it. So that's what I was really encouraging as a starting point for space, because we can do that. You can't do a 50-year humans in space kind of temporal evolution study, but you can do these model organism studies.
I'll just quickly add to that it's always important to keep in mind that we're really talking about two different kinds of genetic information. One is your constitutional DNA, your genome, and then there's also your transcriptome, which is the expression of that. And there are some studies there are databases in the public domain. There's one in particular called NOMAD, which is literally online. It's over 120,000 human exomes or genomes that we have sequenced the nuclear or the constitutional DNA. Some of those people had adult onset diseases, none of them had pediatric onset diseases, and some of them were not appreciated to have any disease. But that database was created to provide us in the genetics community, the human genetics community, sort of a baseline or a reference set to say, if we sequence somebody who's sick and we see a mutation in that person, we can go and look at those 120 or 30,000 individuals as a reference and say, well, how commonly do we see that mutation in this large database? But that's constitutional DNA. The transcriptomics, I think, is a much more complex and tricky question, as we've been hearing people, talking about today, because your transcriptome changes throughout the day in response to many, many stimuli, and it is different in different cell types. And so there is also another online database that's called GTEx. It's the Human Transcriptome, whatever, and it's an attempt to provide to the community some transcriptomic data that was generated, some in mouse and some in human, in different tissues. And that's tissue, that's not cell type. And that's tissue taken at who knows what time of day, and who knows what they ate before you sampled the tissue. So I think with transcriptomics, we have a long way to go with having sort of that reference data set, and your studies will need to be designed with the appropriate controls because of that. When it comes to sequencing human genomes and interpreting that, we do have a pretty decent reference data set for that.
Okay, thank you so much. Okay, final question, comments?
Sorry, I have a poor English, maybe Jessica help me. Okay. 中国方面呢，对这个基因方面研究，包括生命科学很重视。那么专门有团队和办公室的研究。昨天晚上呢，我们这个宇航员第一位宇航员杨利伟先生，还有我们李英威院士啊，也对这个会议的交流很重视。比如说刚才谈到的这个基因的研究啊。那么就提议也很关注，有的问题是有没有可能啊，在一个官方的层面，我们可以有一个合同或者法律的约定，那么对研究的保密和未来的成果可以共享，这是中国方面很关注的。好，谢谢大家。
Now the Chinese attaches high importance to the study of genomics, and we have set up a special joint working team in office in China. And yesterday, Dr. Wang discussed with the first astronaut, Mr. Yang Liwei, and the Chinese academician Li Yinghui, and about this situation with this conference, and shared their opinion. And we have a question: is there an official protocol about the genomic study confidentiality and the data sharing protocol?
Okay. Tom, that might be yours.
I didn't catch the last part of it.
So is there an official policy for data, we'll say repository and sharing? Is there a way to, I think, to maybe collaborate and share data? Is that... Yes.
There's a policy regarding the confidentiality of, and to take part in this data for genomic study and to share the data.
Okay.
So we are.
Okay, Bill. Yeah,
Bill,
actually, let Bill start it. That's okay. Yeah.
We're clearly able to and willing to share any of the data that we've collected from non-human subjects. And so those all exist on this gene lab, and we'll keep populating the gene lab with that. With humans, it's a different question, and for individual sharing, we would have to go through a separate informed consent process with the crew member to be able to share the data.
Okay, thanks very much. All right, I think we'll call it here. Obviously, these guys, this is an incredible brain trust here, and they can't run very fast. So if you have a question you didn't get to ask, rush the stage. But we really want to clear it and get ready for the Apollo event. So thank you guys very much. Sorry.